Thank you for inviting me, Nils. Hi, my name is Eunice. I'm a current fourth year undergrad at Duke University. I've been working at High Dive Lab since this summer to work on building some toolkits that we really can extend us into this 3D space that Eric has mentioned. So today I want to introduce you to a new Python package that we recently just released called InterSCellar. And essentially, this is a surface-based cell neighborhood and interaction volume analysis platform in 3D spatial omics data. So, kind of briefly touching upon what Eric mentioned a little bit before, we want to focus on the surfaces of cells as their avenue of interaction. And this is because cell to cell communication, which is critical in understanding disease pathologies and disease progression, such as the widely well known PD-1, PD-L1 ligand to receptor pair for cancer and tumor suppression, we see that these interactions occur either when cells are directly in contact with each other or in very close proximity with each other in terms of their actual membranes. And when we take that into a larger scale for cell neighborhoods and a more cluster basis rather than just binary, we can see that that can lead up to different disease progressions and be able to differentiate how immune to tumor interactions are happening, et cetera, and really understand the biology behind all of that. And so there's a wider consortia effort, such as through HubMap or Human Cell Atlas, et cetera, that's really looking at this spatial scale and really wants to focus on merging that single cell data that we have with the spatial dimension to further that knowledge and biological discovery. And so looking at those cell neighborhoods, let's say we have like this model of cells in a specific tissue. What does current microscopy techniques do with 2D based analysis? Well, it can kind of slice through a top slice there to look at it from a top-down view, and we come up with this microscopy image of one cell representing this entire cell neighborhood motif. If we slice through a little bit down the z-axis, then we come up with two different cells now here. Slice through there, now the two cells are touching. And if we would go even bottom to the lower z-axis, then we now finally see the different red cells that were not even present in the different slides above. So if we took that to scale and looked at an actual data set, this is actually an Apari view of the exact same cell clusters from melanoma in situ tissues for 2D and 3D space. And if you look at this neighborhood specifically very closely, you can see that the green cell in the 2D space, when translated into the 3D space, is actually touching all of these larger three pink cells, which is not representative in that 2D space because it just seems smaller in size, and the cells that are pink are not even touching each other as neighbors. If you looked at this neighborhood, for instance, then you can see that the two other pink or red cells that are clearly apparent in 3D space does not even show up in 2D, just like the example we showed on the left-hand side. And that one cell that looks a little further apart from the whole cluster in 2D is actually part of the whole cluster in 3D. So the point we really want to drive home here is that a 2D snapshot is barely representative of the actual biology. Now, what's really great is that, just like Eric mentioned, on the data side of 3D spatial omics, we now have these light sheet microscopy or multiplexed immunofluorescence techniques such as CISIF or CODEX, where we're able to see cells and sample them in their full format rather than just their 2D platform, and take those cell surface masks as segmentation tools. Even AI tools for Meta, Segment, etc., are now taking this into a more automated process at scale. And by mapping that onto the different channel protein expressions or even transcriptomics, et cetera, we can map the XYZ coordinates on top of each other to see what cells are there, how they are segmented, and what expressions of biomarkers are present within those spaces, as well as interactively visualize them. And based on that, we can even annotate the different metadata for these cells, such as their types, by looking at what key biomarkers are being expressed and kind of look at their cell states, et cetera. So that was on the data side. What's more on the analysis side here is that we have lots of tools to be able to extract different features from the volumes that we have and the data that we have. And we do so by using more optimized data formats such as Omits or NGFF, which essentially chunks everything down into cubes. And SCVerse is now also trying to integrate this into a more interoperable data format of spatial data. And just like the Nexus paper that we kind of shared earlier on, we're able to also do more network analyses by looking at these visualized cell 3D segments and then see the actual tissue connectivity between them for motif analysis and also interactively visualize that on the test platforms or through different data formats like Neurogrounds pre-computed on Google or even Napari. And all of this now leads us into biological discovery just by being able to have that data sets in 3D and then be able to analyze the data out of it and extract the statistics out of it, which is essentially how we're driving everything into the 3D space further, since biology is simply put 3D. All right, so we got the data side and we now have the analysis side. What we really want to do to bridge these two gaps together is to be able to process the data of actual cell-to-cell interactions within the entire tissue set in a more binary basis. But what tools are currently out there to do this that's both optimized and biologically relevant is currently what we find missing in the field. And here's where we introduce InterSCellar as a Python package that, number one, redefines cell neighbors in a more biologically interpretable way, and number two, computes the interaction spaces between those neighboring cells. And how we do that is that we actually go back to the cell surfaces now that we have the segments available from those 3D volumes and base it on their surface proximity to see which cells are actually neighboring each other. And based on that, really have those interaction spaces between every single neighbor that we find and render them as physical volumes so that people are able to directly extract transcriptomics or proteomics or different multiomic expressions out of them. So let's dive a little deeper into what this package is actually doing. How do we quote unquote redefine cell neighbors? In current 2D analyses, we take a centroid-based approach in literature standard, kind of taking the centroid of this 2D blue cell, for instance, and then doing a radius search of 30 to 50 microns and say whatever falls within that radius will be the neighbor of the cell. But looking at that same representation in 3D space, we see that this method actually misses a lot of cells if we use that radius because the red cell that's not falling into that radius is actually a neighboring cell, inherently because of that sampling bias that 2D brings us. Then you could argue that you could use a larger radius to capture more cells, but now the problem is that the radius is now capturing all of the cells on the bottom since it's that circular shape and kind of constrained into that geometry, so that cells that are not actually touching each other and actually has different cells in between each other are now also being considered as neighbors, which is simply inaccurate. And so what InterSCellar wants to do is that it wants to segment or take the segmentations of existing cells and dilate that surface directly following its shape, not in a circular format or a spherical format, but directly just dilating orthogonally from the cell membranes. And whatever cell and whatever the user defines that distance threshold to be, the cells that are touching right next to each other or very close in that proximity will be considered as neighbors. So the user could either query through their entire tissue data set and say, I just want to find touching neighbors with shared surfaces because the biology of my data sets and refers to that as what cells can be communicating with each other. But in addition to that, if the user wanted to say, oh, I also want to look at neighbors that are quite close to each other, maybe within a 2 micron distance, but not necessarily sharing a touching surface, that is also queried through, depending on what the user defines on InterSCellar, through a Euclidean distance transform directly from the cell membrane in order to see what's kind of in that vicinity when you're taking the shortest distance from every cell membrane to each other. And once you find all of those new cell neighbors, we want to store this into a cell neighbor node edge graph so that users can simply query and optimize their search process throughout the entire data set by looking at those unique pairs with their metadata and relationships stored into them. So for instance, a user could query all neighbors of one very specific cell they want to look at and look at that cell neighbor cluster, since we have this data form available on AND data as well. Or they could even query different types or a very specific interaction type, such as tumor to CD8 T cell pairs, but across the entire tissue data set so that you can visualize and kind of pull out metrics for that specific pairing and how that interaction looks like in the entire space of the data set you have. So now that we have those cell neighborhoods, we want to compute the interaction space as physical volumes for every single neighboring pair that we have found and have stored into this graph. So how do we do this? We take a pretty geometric approach here, but to break it down, essentially, we're going to call this interaction space where cell signaling occurs and call that as our intercellular volume. What the intercellular volume consists of is first the intercellular volume, so kind of like the extracellular matrix between two cells where cell signaling can be occurring, where proteins or genes transcriptomics can be being passed across each other. Add that to the intracellular volume by dilating that inwards within the cell membrane, since the space that is very close to the cell surface can be irrelevant as to what markers exist and what is being communicated across each other, and also the contact surface between two cells if they are in contact. And once we compute all of that, we're going to subtract any overlaps with third-party cells since we want to consider just the binary interaction for every single cell pair neighbor. And that dilation in both the intercellular and intracellular sense is going to happen depending on what the user defines as how far they want to kind of go into. So depending on what maximum distance threshold they choose to go, the algorithm essentially looks at the specific background voxels between each cell pair and then takes the shortest point to surface distance, adds them up, and if that falls under whatever the user defines as a distance threshold, that's going to be counted in as the intercellular volume, and then dilate that inwards to kind of find the intracellular volume. And since the user can define how far that distance is going to be, maybe 1 to 2 microns, that's going to be very adjustable to the biology of the tissue since every cell type kind of interacts in a different way, and that distance might also differ. So this white space is kind of what you could visualize as that intercellular volume between two cells. This is an Apari view of then what we see. Once we have the intercellular volume computed for all these neighbors, we also can subtract out the cell volumes from the intercellular volume from the original cell segmentations to find the cell-only volumes, excluding that intercellular space. And what essentially intercellular takes as an output, as our final output, is that it gives two global images in 3D volume format where querying through the entire tissue map data set, you have all those different neighbors. So you have all the volumes of intercellular volume, all the intercellular volumes in a singular OmiSAR data frame, as well as all the cell-only volumes so that you could interactively visualize that and query that through in the graph. I do want to briefly mention that our package also does support a 2D feature as a parallel arm, as we do acknowledge that there is a vast amount of 2D data sets out there, at least as of this stage, more so than 3D. So it kind of takes the same approach where you segment out the cells and then the algorithm essentially dilates it in an orthogonal manner to see what cells are kind of neighboring each other, where the user can define their distance threshold to kind of adjust how much of a bandwidth they want to go into that dilation. And you come up with this final image of the cell-only volumes and intercellular volumes together, where those are essentially the intercellular areas, essentially the in-between spaces between the 2D cells, and those are the cell-only areas, so they can kind of map onto each other. So kind of just to test out what this package would look like in real life, we collaborated with Peter Swerger's group in order to look at melanoma in situ in CISIV 3D datasets. And our input here was around 11K cells. And we kind of set these different thresholds to see, oh, I want to search all these cell neighbors that are within a proximity of 0.5 microns from each other, and then try to compute the intercellular volume between them and intracellular volume between them to come up with a finer intercellular volume. And this is kind of what we had as our output for everything together with the cells and all of their intercellular volumes. What you see on the right side here is just the intercellular volumes between the cells, and we were able to identify around 45,000 cell neighbors from this data set and compute intercellular volumes for that, where around 26,000 were touching neighbors and 19,000 were near neighbors. And for 13 different cell types that were present in the data, intercellular essentially found around 70 unique cell-to-cell type pairs. So this would be the cell-only volumes kind of subtracting out that intercellular volume from the original segmentation. And you see a fuller picture here with all of those volumes in between. And now you have everything segmented out as essentially voxel data frames where you can do feature extraction in order to see what biomarkers are present in that space. As a quick overview of like the runtime efficiency of this whole method, I ran this fully locally on my computer for 4 CPU cores kind of parallelized onto each other. And this full pipeline for around 10K cells took around less than 2 hours to both find the neighbors and also compute all of these volume spaces. And kind of going from here, it's a very exploratory route for the biologist in terms of the discovery side, as now you can see the different expression levels for every single channel you have in that intercellular space and query it by different cell-cell pair types or interaction pairs. You could also zoom into one of these very specific proteins or transcriptomes and kind of see that probability density of what their expression looks like over the scale for both cell A and cell B. So for instance, like a tumor cell and a CD8 T cell inside those volumes, and then as well as the intercellular volumes. So that's kind of where InterSCellar is at right now. For future works, we understand that there are greater intricacies with cell-to-cell interaction, such as polarization of biomarker expression within these spaces, as well as different geometries of cell surfaces that can be affecting the way that cells are communicating with each other. So not just their proximity, but also their texture, how cells are junctioned with each other. Is it smooth? Is it kind of like a knuckle feature? Things like that. And we also want to make the platform a little more interoperable with different data frames. Currently, we're supporting a lot of de facto standardized data formats such as OMI-ZAR, OMI-NGFF, as well as collaborating with a CVerse for more and data, mu data, spatial data interactiveness. But we also want to be able to take that into the test and really visualize it on a more on-demand manner on a web-based basis, and also try to have it more seamlessly transition into feature extraction tools that are externally existing for these volumes that we create. And we're also looking for more data sets to test on, especially for 3D in terms of maybe transcriptomics or metabolomics in order to test the biological validity and what that story can really bring us through InterSCellar. So with that, I just really want to acknowledge our team. I really want to thank my PI Nils and my mentor Eric for really helping with this process, and also our collaborators at LSP, Peter Zwerger's lab, Clarence Alton and Alex have been wonderful, providing some biological insights into these methods, and also our spatial biology team within the lab. Yeah, thank you. And with that, I'm happy to take any questions.
Thank you, Eunice, for a great presentation. Very exciting work.